Hello everybody, welcome you all in the webinar. The topic I am going to present is the overview on applications of bioinformatics in the COVID-19. Myself Subhamoy Banerjee, I am an assistant professor in School of Life Science, Crescent Institute. My research interest is viral genomics and molecular comorbidity analysis. Today we are going to discuss about the bioinformatic application about the COVID-19. So, at a glance, this COVID-19 is caused by SARS coronavirus 2. Sometimes it is called a novel coronavirus 2019. It belongs to beta coronavirus family. It is a single-stranded positive sense RNA genome. The size comes around 29 to 31 kb, and it was first reported in November 2019 in Wuhan, China, and this is a large genome, so it has almost 20 protein encoding genes. So there are 16 non-structural and 4 structural proteins encoded by the SARS-CoV-2 genome. So, according to the latest update by 17.5 in Worldometer, the coronavirus cases there are nearly 50 lakh. It's 47 lakh 50,000. The death is above 3 lakh, and since till now the recovery is the 18 lakh 30,000 is the global rate. But according to the estimation, we have not reached the peak yet, and for many countries it's just a beginning of the log phase. So many more cases, number of cases and deaths we can expect in the near future. So the before while we deal with this viral genomics, so computational viral genomics, some of the basic concepts which has already you have been through in previous two lectures, the BLAST, multiple sequence alignment, and phylogenetic analysis. So as you have seen that this multiple sequence alignment and phylogenetic analysis, they have different algorithms. So the depending on the type of viral sequences and many other parameters, you need to choose what kind of multiple sequence alignment algorithm or the program you are going to utilize, and the what type of phylogenetic analysis you are going to employ. So the databases from which these coronavirus sequences are downloaded, majorly the NCBI virus database, then GISAID, and the Viper. So these are the major. There are many more small databases are there. So in this here we'll see that if you want to do just as a very rudimentary level of bioinformatics analysis for this coronavirus, so what method you can apply. So for example, you are going to take the go to the corona NCBI virus database. There you will find that the SARS-CoV-2 database. From there you have two options, nucleotide and protein. So you can select either of it. For me, I selected the nucleotide. So now for the BLAST, you can run one sequence, or if you want to check the multiple sequence, you have to go for the you can select more than one sequence, and then you can construct a phylogenetic tree in order to see the evolutionary relationship. So here is the example. So the first example is that this is a screenshot where I have you see that coronavirus data hub. I selected the complete genome. So in that complete genome, there are total till now globally 3,863 complete genome has been submitted. So in that complete genome, as I selected India, so there till now total 78 sequencing has been done. So you can see that majority of this has been from the Ahmedabad, and there are many more sequences are there. So from these sequences, you see that, as I told, if you want to do the BLAST analysis, so what you have to do, you can select any one of the sequence, suppose this one, and you are going to download the FASTA sequence from there. And as you have seen in previous lecture, that how you can run the BLAST. So naturally, once you have the FASTA sequence, you can go to the nucleotide BLAST or some other specialized BLAST, and you can check that what is the nearest sequence in the database available by running the BLAST. So but here what I have done is I wanted to go for a multiple sequence alignment, so I selected all the sequences, and after I selected all the sequences, I ran the MSA here in the NCBI itself. Otherwise, I could have downloaded the multiple FASTA file, and I could have run it in some other database, other websites like ClusterW, EBI tools, which I could have used to run in the ClusterW. But anyway, here I just ran it, and here I have you can see that you are seeing the multiple sequence alignment result, where some matches and the mismatches has been mentioned here. So you have the file that is a .aln file. So you can directly go for the build phylogenetic tree also from here, but I took a different approach. So I went to the phylogeny.fr. So they have a new site that is called nphylogeny.fr, and there I I ran the phylif. I converted it into the phylif file and I ran it. So here the linear comes actually quite big, which will not fit in the window, so that's why I showed it in a radial fashion. So anything is possible. So this is a phylogram where you see the length is specifically it is showing the length to the scale bar is like this 0.001. So actually it indicates that there is not much difference among the strengths. And then so actually not much difference, I mean to say that there is a very minor changes among the one strand to another strand. So then I check the dendrogram. The dendrogram is basically the hierarchical clustering is done in the dendrogram analysis, so that there it is coming like this. This is a phylogenetic analysis, and from there I mean many more complex phylogenetic analysis can be done with the help of the Bayesian algorithm. So there you so etc. So there you need the numeric file. So the file format, the alignment file format varies from what type of phylogenetic analysis you are going to uh, employ. So here we'll see some published data what they have done in the bioinformatic analysis. So here in this Lancet paper, so this was the around February that paper came. So actually in the bioinformatic analysis you will see that the more number of sequences are coming, so the type of data is also changing. So this is the initial where they isolated some of the coronavirus samples from the patient, and they have found gone for a multiple sequence alignment first. Here you see that the synonymous and non-synonymous mutations they found out, and after that, after finding out that non-synonymous this mutations, so they went for the further the structure, the protein structure. They try to check that what are the different changes, and spike protein is always a matter of interest for the coronavirus, because they use spike protein in order to access our epithelial cells. So here majorly they have taken the coronavirus, the bat coronavirus, which is actually most closely related to this novel coronavirus, and the SARS, which came around 15-16 years back, and the MERS, which came around 2012-13. So these are the sequences they have taken the data they have taken to go for a multiple sequence alignment to identify that where exactly the changes are there. So this is the cladogram or clade-based analysis. So there is a different substrains of SARS-CoV which is present in the database. Then bat CoV, the different type of bat CoV they have already identified, and then the novel coronavirus, and then the few other. So that you see that the bat CoV is also there is a different clades there, and then Mars. And one thing you can see that the novel coronavirus, by the bioinformatic analysis, it was first identified that this novel coronavirus is actually much more closely related towards the bat coronavirus as compared to the SARS or MERS. So the whole approach, I mean the therapeutic approach needs to be revisited. So that was the major idea that we got from the this bioinformatic analysis, initial bioinformatic analysis. So another phylodynamic analysis which was posted on the 6th March. Here actually the amount of data is coming every day. So this dates of the publications are becoming very important. So here this one scientist, Professor Andrew Rambot. So he showed that when he took these different viral samples, the different data from different countries, he showed that how the changes, how they are varying from one strain to another strain. So you see that one paper which came in February, so maybe up to the January with the data set, and another paper that is another work that is coming on the March. You see that there is a huge variation we can see in the phylogram. And so in another analysis to estimate the date of the most recent common ancestor, the author has used the Bayesian evolutionary analysis of sampling tree. The software is named as Beast, and he identified the estimated the date which is corresponding to the the different different reports about that first reported the origins of the first reported case related to the corona. So in another bioinformatics work, authors tried to identify the but coronavirus why the coronavirus is affecting some parts of the world more than the some other part of the world. So here they have identified that there are 11 clades are there among the since its origin. The origin is was considered as the O clade, where it was much higher in China, and they found that this in India there are the out of the 26 clades what they identified until their publication, they found that this A3 and A2A clade was more dominant, and then they went on analyzing that so how these clades actually moved, how these clades actually changed. So in the East Asia, it was shown that this initially, before just going to before going analyzing this black and this red and blue, I'll just show you that what they found is that in the spike protein at the 614 position of the amino acid, the alanine is changed into the glycine. So according to their analysis, this change actually leads towards the elastase, which is a sedent protease that can cleave this this protein. And so now, so that is that mutation here they have shown. So the in East Asia, in Europe and in East Asia, actually it was shown that slowly this variant is taking over the other variants. So the authors identified that the single mutation at the 614 position, these strains quickly outcompeted the original strain all across the globe. And they also found that a specific deletion mutation in the TMPRSS2 gene, which is common among the Europeans and North Americans but extremely rare among the East Asians human population, actually helped in the entry of this particular strain and this coronavirus strain into their body. So this is a website that names the next trend. So they do a real time tracking of the pathogen evolution. So the next comes the phylogenetic network analysis of SARS-CoV-2 genome. So this is one approach that is called a median joining network approach. So the authors had done to identify the how they took 160 different strains, and they identified that how these strains are joined with each other. And and their conclusion in this work was that the network faithfully traces roots of the infection for documented coronavirus cases, indicating the phylogenetic network can likely be successfully used to help to trace the undocumented COVID-19 infection sources, which can then be quarantined to prevent the recurrent spread of disease worldwide. So this paper came in the PNS. Now, we should be little careful while doing the evolutionary analysis. The reason is that against that paper, the rebuttal came up from different sources that how the sampling bias and incorrect routing make the phylogenetic network tracing of SARS-CoV-2 infection unreliable. Another letter to the editor suggests that the median joining network analysis of SARS-CoV-2 genome is neither phylogenetic nor evolutionary. So, while doing this kind of evolutionary routing and analysis with the help of bioinformatics, we should be little careful about the choice and the other related and the whole process. So the another approach used in bioinformatics is called the drug repurposing. So the drug repurposing is a strategy where a discovery of the new set of drugs from the existing repertoire of the drugs. In the pandemic situation, especially here we are going through now, so they are finding out a novel therapeutic agent is time consuming, and also it takes a lot of the effort, and by in the meantime supporting the patients is a crucial matter. So there the drug repurposing comes into the play. So here what the authors have done is that they've identified the protein subnetworks, how the these coronavirus proteins are affecting the human proteins and network interact. So then they pull the from the human interactome. They find out that how the human interactome actually how the one protein is interacting with the another protein can be is identified. So they are here how this coronavirus proteins are interacting with the human proteins were actually pulled, and then the drug target network was identified, which proteins are can be targetable. So next is that they found out the proper targets whom the coronavirus or which proteins in the interactome the coronavirus is affecting, and which proteins are affected by the drug, which finally would help the coronavirus to be ineffective. So from there, they have used that further they gone for the transcriptome analysis, and then they enriched, and after the enrichment they made a network based prediction of the drug combinations here. So they found nearly three drug combination may play a very important role in the coronavirus treatment. So in another work, the authors screened the ligand library for the 61 reported antiviral agents, and then they found out the crystal structure of the target proteins. They went for the docking and screened the most important hits. So which drugs can work usefully against the coronavirus? In another this type of approach, the authors have identified have screened the zinc database for almost 30,000 molecules, and drug database. From there, they went for a virtual screening with the help of Autodoc Vina, and there they took a threshold binding energy threshold of -8 kilocalorie per mole, and from there they actually screened it. So the screening suggested that the uh, much low so the binding energy which are lower than the -8, though they are taken for the molecular docking, and then they tried to identify the best hits, and they found the screening result. So the another important approach comes is the PCR primer designing. So there are several softwares are there like Primer 3, NCBI Primer Blast, etc., where the PCR primer, specifically the real-time PCR primer, and the loop mediated amplification, isothermal loop mediated amplification, those primers are designed. So they are the knowledge of uh, sequence is necessary in order to specifically to design the primer which is specific to the coronavirus, to maybe any subtypes, and it definitely it should not give any it should not bind with the host. So in this paper they actually identified the WHO listed qPCR primers, and went for a in silico PCR, and then they found that some of the Chinese CDC engine specific qPCR primers are mispriming. So here also the necessary filtering should be done for the to design the primers. So altogether in this webinar, I tried to give you an overview about that how the different ways the bioinformatics can play a very important role in order to fight the COVID-19. So thank you everyone for registering with us in this webinar.